All right, welcome back, everyone. We're gonna kick off here, and so we have a little time for questions and Q&A and discussion. All right, round two, calculating DNA properties. Where did I set? There we go. I'm gonna get my pointer. All right, so we talked a little bit about DNA, what it is. I give you a little bit of insight of why we should study it, right? Cancer research, cancer therapeutics, understanding human physiology, understanding inflammation in the heart pathways. All kinds of stuff, right? But how do we do it? What are the tools that we're going to use? All right. So, big surprise, DNA, you can measure its concentration. Right? So what's concentration? What does the term mean? Fundamentally. Amount per volume, right? How much is in the solution? So who here, raise their hands, who here has worked with DNA before? All right, has anyone ordered DNA primers? Yes? Right? Comes in a tube. You look at the tube and you're like, man, did I just get ripped off? There's nothing in there. Right? You can't see it. Now, when I was in my PhD days, we made enough of it because you're slave labor sometimes, and you could see it as a giant fluffy cotton ball. Right? There's a whole lot of DNA. But how do we measure the concentration of something we can't see, something you can't taste, something you can't touch, you can't feel, right? I guess if you had as much as I had in a cotton ball, it might have tasted sweet. You might know something, but right, there are some sugars. What are we going to use? What are we measuring here? Absorbance. Absorbance, right? So DNA base pairs have an extinction coefficient roughly of about 10,000 per base pair. Okay? For double-stranded plasmid, we can measure them at 260 nanometers. You can measure it in micrograms per mil, right? I always go back to concentration. If you do crystallography, you're going to typically be in grams per liter or micrograms per amount, right? For NMR, we often work in molarity, okay? 260, the actual peak is 254, 260 is close enough, right? What's the absorbance of protein? Who knows? 280, yes, 280. 280 is protein, 260 is DNA. Where do these numbers come from? Is it magic from the sky? Someone just said 280 works. Hint, what is this? It's an absorbance, it's a scan, right? If we take our spectrometer, and all we're doing is we're scanning. We shine some light into our sample, and we scan the wavelength. And this is the amplitude, right? This is how much is coming back. Absorbance is related to percent transmittance, right? We can see how much is absorbed by how much light makes it through. So you can look at different concentrations of DNA. What's really nice, you can look at very dilute concentrations of DNA because it's 10,000. The extinction coefficient is 10,000. What units is an extinction coefficient? Okay, what governs this rule? How do we get from absorbance to concentration? Beer's law, the drunken alphabet, right? Back in high school when I was a teacher, we called it the drunken alphabet. A equals EBC. It's corny, but you won't forget it, right? A is absorbance, E is epsilon or extinction coefficient, B is path length, and C is concentration. In what units? Concentration in molarity, right? Moles per liter. Path length is in centimeters. If you visualize the equation or if you write it down, A equals EBC. If I take A and I divide it by BC, A is unitless. I get 1 over centimeters, inverse concentration, right? So 1 over inverse centimeters, inverse molarity. If you do not understand units, you will not calculate the correct thing. You will have very wacky numbers and waste your boss's resources and your time. Pay attention to units. Always pay attention to units. Pay attention to big picture and units. Those are what matter. Okay, so we can do these scans and we can add more DNA and we can watch the concentration go up. DNA is a double strand. We said 0.2. Protein, 280. So GB3 is a protein they work on in the Fitzke laboratory. There's an extinction coefficient of 0.0016 micrograms per mil. Where did the extinction coefficients from proteins come, and where did the extinction coefficients come from DNA? Why do they absorb? What type of structures absorb energy in this region, this frequency? Pi electrons. What do those look like? Rings. We're looking for rings, right? Nucleic acids, we talked about purines and pyrimidines, they have ring systems, right? Ring systems are great. So what amino acids contribute to your extinction coefficient? What's the best one? Tryptophan. What's next? Tyrosine. And what's last? Phenylalanine. Yep. Cysteine, watch out for it, doesn't contribute, but if you have disulfide bonding, it can get weird, right? All right, so tryptophans, tyrosines, phenylalanines. If you're fortunate enough to have a tryptophan, I'm usually not. That's awesome. Life is good because it's about 5,000. If you have a tyrosine, it's 1,490. If you have a phenylalanine, well, life kind of stinks because it's 180, right? We can use phenylalanine for certain parts of this protein calmodulin. We measure the end domain with phenylalanine. It's not fun. Any impurity, any trace amount of any other protein with a tyrosine, and we're way off, right? So we have to do other things. So which is more sensitive? This one or this one? Which one can you measure a smaller concentration? DNA or the Fitzke Lab GB3 protein? DNA. Why? Think through it, right? You guys are scientists. Over here, I get more bang for my buck, right? For each molecule of this, I contribute this much to the absorbance. For each molecule here, I contribute, well, not each molecule, I get a different amount, a lower amount to the absorbance. So if you could think of it as noise, right? If you want sound, each molecule yells louder. Therefore, you can detect smaller amounts. All right? What does that mean? You can't work it at smaller concentrations, or your precision of your measurements may be sacrificed. All right, so Beer's law has some implications to it, right? A equals EBC. Is it always true? Does it fail us? Yes, I see a head nodding, yes. Pay attention to this classic first-time mistake you get in a research laboratory. Your spectrometer may be the best spectrometer on the planet, but it's still a subject to Beer's law. Where do you have to measure things? Between what ranges? 0.1 and 1, I argue, I always say 0.3 and 1.6 if your spectrometer is made for it. Okay? The reason for that is percent transmittance. How's it related to Beer's law, to absorbance? It's inverse, but what type of mathematical function gets you from percent transmittance to absorbance? Logarithmic, thank you. In a logarithmic curve, if I draw it, arches like this. And all we're doing with Beer's law is we're fitting a linear line between this range. Here, you're massively undersensitive. Here, you're massively oversensitive, right? And so you introduce error. So when we measure UV-vis absorbance, you need to be between 0.3. Some spectrometers cut off at 1, so you have to know your instrument. If you work with a carry 3, you can go up to 2. If you work with a Shimatsu, they're usually at 1. Depends what you're doing. And always work with, if you can, a path length of 1 centimeter. Those tiny little cuvettes that are black that have the windows shaded in, only if necessary. Make sure you talk to your boss before you do it, right? They often have errors. Path lengths get funky, especially with air bubbles. All right. So long randomized sequences, single strands, we have a certain extinction coefficient, right? Single strand DNA, a different one. Remember we talked about base pairing stacking. When you create order, you stack the pi to pi star transition, the different pi stackings changes. You have something called hyperchromicity and hypochromicity. It's how we measure DNA melting, right? As it comes apart, the DNA unstacks and you actually get an increase in the absorbance, right? And we can measure that, right? You can look at ratios based on the wavelength. We can learn something about samples. So now you go to isolate, you put your plasmid in, you did all your molecular biology for 6 months, you're overexpressing an E. coli, you purify your protein, you want to know how much DNA contaminant you have. You can look at your absorption spectra. And you can see from UV-vis, do I have some nucleic acid at 260? What's the third wavelength? Anyone know that's of use for usually for overexpression that we monitor? 260 is nucleic acid, 280 is protein. What's the other one? Anyone know? Say again? No. 214 is very helpful. 214 tells you an amide backbone bond, right? So if you have a protein that doesn't have an extinction coefficient, say you're purifying on something like HPLC, you can monitor 214 to look for your peak. So nucleic acids and small molecules. So we have an Oligo Analyzer. Has everyone loaded this and played with this a little bit? Is this from IDT? Yes, IDT. IDT, Integrated DNA Technology, right? So you can look this up online. They update this every couple of years, different flavor of it. We can calculate properties or predict them based on what we know about nucleic acids. So you can load in your sequence here. We can set our parameters. We can have it analyzed. We can calculate things such as what's the propensity to form a hairpin, right? What's a hairpin? Not the thing that goes in your hair, but what's a nucleic acid hairpin? Exactly. It can loop around and bind to itself, right? We can have A go to T. We can have G go to C. These ends are pre-localized to each other. In solution, it's floppy, right? And it may want to go like this and fold back on itself. Why do you care about that? Can someone give me an example? You're working with your molecular biology, right? And your boss says, make this mutation in our protein and tell me if it makes it less stable or more stable. So now you have to go figure out how to mutate a protein. So you look up quick change mutagenesis, right? And you got to do something called design a primer, right? And that primer needs to bind in a DNA duplex, not in a hairpin. If you design your primer so you get hairpin, will you make your mutation? No. No. For those who aren't familiar with quick change, we take a small piece of DNA, we use it as tape. We make a specific intentional mismatch in it. We lay it down on top of the plasmid, we anneal it, and we have an enzyme come along, and when it hits that bubble, it actually incorporates the wrong amino acid on purpose, right? If you're doing quick change, use an enzyme that doesn't have proofreading capabilities, right? If you have proofreading capabilities, it'll fix the error, not make your mutation. Anyway, things that'll come back to bite you. So we can calculate these things, right? You can log in, and has everyone had a chance to log into the screen, take a look at this? Yes, no, maybe, if you're... All right. You can calculate a reverse complement, right? Now, 5' to 3', right? That's what we said. There's directionality on your nucleic acid. You can write out by hand reverse complements. My boss tortured me for years and made us do all this by hand. There's this wonderful little thing. You can click reverse complement. It will give you the answer, right? You have to remember it goes 5' to 3'. You can make the opposite, but you have to flip around the direction. You have to reverse the sequence and then take the complement. You can't do one or the other. All right. You can translate DNA, right? Most of these things, if you want to use something outside of IDT, XPC is wonderful. This is mostly free. You can log on. It has all kinds of interesting little tools, right? There's a translate program that will tell you what reading frames there are, what your gene will produce. If you make a mutation in a plasmid for someone, please be a kind scientist and sequence your plasmid afterwards to confirm you made the right plasmid before you send it to your collaborator. It will not endear you to your collaborator, I promise. NCBI. What's NCBI? You're right, but you got to say that a whole lot louder. National Center for Biotechnology Institute, right? So who pays for this? We do. It's our tax dollars, right? Federal government sponsors this. We have a database which allows us to work with sequences. So you're already paying for it, so you should check it out and use it, okay? Don't sit there and try and calculate everything by hand. I've had people take the little table and try and translate everything on their own, do reverse complements. We already have tools. Use them, play with them. They're faster, right? Use them to double check yourself. It's a good deal. PDB, what does that stand for? I'll give you a hint. P stands for protein. Protein data bank. This was a huge turning point in my career. When I found this, you can download structures of proteins, of nucleic acids that have been determined by other scientists and play with them, look at them, explore them, make amazing artistic figures out of them. Look at how they interact. You actually can look at the atoms in space and see where they are and make predictions about what's going to happen. So when you embark upon your research projects and you don't know something, don't just blindly be like, well, we're going to make it and make this measurement. Look up the proteins, look up the structures of the things you're working with. Right? Science isn't something you do 9 to 5. It's something that you get addicted to and it becomes your lifestyle. So you can understand how the PDB houses a ton of DNA and RNA structures, right? And you can look at how all of these interact. You can play with programs like VMD, PyMol, Chimera. You can do all sorts of amazing stuff, right? How can they make your lab work easier? What have we talked about? Okay, let's assume someone comes to me and says, Dr. Johnson, I want to do research in the heart field. And I really want to make a difference. I want to become a cardiac surgeon, but I know I want to do research as well. Rather than telling them they're crazy, ask them, well, what have you read about? What have you looked about? Right? Before you dive into a project, the onus is on you to set the background, set the stage, figure out your interests. Before you start using time and resources from a faculty member, the onus is on you to understand what you want to work on, to have an idea, to start to get some traction, right? We can either hold the book for you and you can read it, or you could read that and then come to us and we connect the dots. And that makes life a lot more fun for everyone, right? So these are resources to help build your background. What's super cool, they're free, right? So play with them. So you can study, you can look at structures of proteins you're interested in. You could design some mutations that you think might be interesting based on those primers, right? If some of my friends for Nashville were here, they'd be all about these databases for looking at sick people and which proteins are different, which ones have missense mutations, and whether or not they cause disease. Nowadays, we sequence everyone's DNA. You come in the hospital in Vanderbilt, right? You go in that hospital, unless you specifically opt out and you have blood drawn, we're going to sequence your DNA. And we're going to blind it. And you're going to go in a databank, the exact databank for a healthy individual, unless you have a problem. Then you go in the sick person databank. Because we want to know which mutations in your body you can tolerate and stay healthy. Because there might be someone out there that's deathly ill that has that mutation. But if you're fine with it, then it's not the cause, right? So trying to understand those things. You can play around with that. Get familiar with it. Understand it. DNA melting, we've talked a little bit about this, I think, a couple times, right? You have specific base pairs that come together. Now, this assumes a very simplified system. Remember, I told you G's can pair with T's. It happens. We get these mismatches, right? This assumes we just have a duplex. We have double-stranded. And if you're fortunate enough to have a simplified system, you can calculate a Gibbs free energy. How's that related? Who knows some equations for delta G? Delta H minus T delta S, right? How about another one? Yes, natural. And what's K? Equilibrium, which describes products over reactants, right? You got it. Concentrations of things, right? Awesome. E to the negative RT ln of K, right? And so you can fit these parameters if you understand a little bit about when it's single-stranded, when it's double-stranded. You can calculate some thermodynamic properties, right? Some energetics, and gain some insights. And again, these are important. They allow us to describe a system, right? They allow you to scientifically capture precision in details to compare things back and forth. If we add heat, we're putting energy into a system, right? What happens to nucleic acids? We already talked about it a little. The DNA starts to separates. We call that melting, right? Is it more ordered when it's a duplex or less ordered? More ordered is a duplex, right? We add energy to the system, falls apart, right? What do we call that measure of disorder? Entropy. Entropy wants to increase. All right? Higher temperatures, DNA separates. Right? When you lose this pairing and you have random coil, right? Things are unstructured. They flop around a lot in solution. Remember those 5 torsion angles can do lots of cool things. Right? Primary is maintained. Why? Because primary is driven by sequence. You got it. Now, what's going to affect our melting temperature? Salt, absolutely. Absolutely. Salt's going to change some of the electrostatic charges. That's going to dictate at what temperature you anneal. Right? Do you think sequence is going to influence the melting temperature? How many bonds from an AT? How many bonds from a GC? So if I have just a pure GC strand, will it take more or less energy to melt compared to an AT strand? Take more. You have more bonds to rupture, right? We can calculate that. What's super cool, you can do some math with pencil and paper, and we can go put that in an instrument and measure the physical properties and check your math and see if it's real. To me, that's really cool. Blows my mind. All right. TM. So a TM is the melting temperature. How do we pick which point to define as a TM? So when we melt this, it gives you a classic sigmoidal curve, right? So at the bottom, we're almost all duplex. And at the top, we're almost all single strand. In the middle, you have a slope. So has anyone done any pharmacology? Yes? No? Toxicology? No? Okay. In pharmacology, we love to use a dose-response curve. Okay? Dose-response fitting means there's no assumptions. And what we do is we capture what we call an EC50, or the effective concentration to get halfway. And this is true for most sigmoidal-shaped curves. If we take away all assumptions, at its truest sense, the inflection point, or the second derivative, right? If you've done calculus, you have your first derivative, that's the steepest part. If you take the second derivative, set it equal to 0. So you go from concave up to concave down. This is the steepest slope of the whole chart, right? And therefore the most sensitive, and therefore the most accurate descriptor. So your melting temperature is at your inflection point. And that's where you have 50% DNA duplex, 50% single strand, right? Same is true if I'm administering a small molecule to a mouse and I want to know the EC50, right? The most sensitive point is at the inflection point. Same is true if I give a calcium to a calcium binding protein. As long as it fits a dose response curve, EC50 is similar to a TM. All right. Now, if we calculate a TM, add 4 degrees for each GC base pair, 2 Celsius for AT. It's an approximation, right? You can predict it's at 70. All right. You can get a little more sophisticated. You can calculate some of these with the IDT Oligo Analyzer. Remember, this assumes two-state system, right? It's all duplexed. It's all single-stranded. It doesn't take into account other forms, mismatches, hairpins, other types of tertiary and quaternary structure. If you work with small pieces that are less than 10 to 15 base pairs, you'll be fine. When you get longer, it gets funky. Right? All right, so you need DNA sequence. As we said earlier, we need a salt concentration. And it's going to tell you what the melting temperature is. It's going to give you an extinction coefficient. It'll tell you the percent GC content. Right? Look at this. We can get really cool structures. We can get these loops and these hairpins forming inside a single strand. Right? Why would you care about all this? Why is this important? Go back to our making a mutation sample, right? You want to make a primer to mutate a sequence in a protein so you can characterize it. If it does this, and this is more stable than laying down in a duplex, is this good? Not if you want to make a mutation, right? So understanding different types of secondary structure and when it's going to be folded correctly or base pairing to what is important. It's what allows us to control molecular biology. Okay, so we have some HIV RNA. Okay, so transactivation response element, and it binds to a TAP protein, which is viral transcription. Here's your sequence. Okay, notice all the U's. This is an RNA this time, right? Why is all this useful? We've been harping on site-directed mutagenesis, right? You got to be able to design primers. So most biochemistry labs are going to study proteins or RNA or DNA. You're going to have to be able to work with this stuff, right? And again, you need to learn it. You don't get points on the board for the molecular biology. That just gets you to make the sample so you can start to characterize it in most cases. Here we have a Nobel Prize in '93 for contributions to the methods of DNA-based chemistry, right? So development of PCR. What is PCR? Polymerized chain reaction, right? Very common technique you will work with when you first come into a research laboratory, right? PCR, it's a thermal cycler, right? So they figured out that if we heat up some enzymes, they work at certain temperatures within a specific range. If we heat them up, we can allow the DNA to anneal, we can turn the enzyme off, we can cool it down, we can let it replicate, come apart, and make another copy. And therefore, you can amplify over and over and make more of your DNA plasmids. Right? So you guys will become familiar most likely with mini preps in molecular biology. Here we go. So we can add our primers together. We can get different copies. We can make 4 copies. We can keep going as we cycle until we have millions and millions of copies. All right? In this manner, if you make your mutant plasmid for me, I can then tell you, hey, you need 1 microgram per batch of cells we're going to transfect. Right? So we do transformations when we put DNA into bacteria. If we're doing transfections, we're putting them into other cell lines. So I do something called whole cell patch clamp electrophysiology. It's a mouthful. We basically take a cell, we artificially insert a plasmid into it, and we make it express an ion channel. And we take a small capillary tube that we pull about as thin as a piece of hair, we attach it and make a circuit. We can manipulate voltages, and I can make those ion channels open and close, and I can characterize how they do that. So when I build structures of them, I can understand, hey, if I bind this protein to it, how does it change the opening and closing? How does that change electrical currents on your cell? Ultimately, how does that interplay to calcium, and how does that change how your heart beats? Right? But I can't do that if we can't make constructs to study. Right? So that's where you guys develop your skills to make all these things, and we can come work together. All right, so designing primers. There are a gazillion resources on designing primers. Use the established protocols. I can't stress this enough. Don't run around and follow what your friend did because the chicken with the head game didn't work well. Does everyone know what I'm saying? If I play telephone and I tell Professor Fitzke something and he passes that information around the room, I can share that I ate a banana here with Professor Fitzke. By the time we get to the back of the room, it's, Dr. Johnson's going to California. The message changes. Use established methodology for designing primers. Do not wing it. Do not go at it. Do not reinvent the wheel. Use the resources available. They're fairly straightforward. All right. Does everyone have a chance to log into this and we can go through an example of it? Yes? No? Maybe? All right. So we get a screen like this and it says we can load in, recommended you clear everything, right? So we can load quick change too. So this is a kit that you buy. They will sell you. You can make all the ingredients if you want to spend your more time and money. You can put in your sequence, right? You can tell which region you want it to do, which nucleotides you'd like to change, and it will come back and tell you which primers to use, right? Good idea to understand what it's doing to double check it, but a great first pass. See down here, we can get our primer template based on which ones you want to choose, how to make mutations. Okay. So some general principles in doing quick change, right? Your GC content should be between 40 and 60%. What happens if it's more than that? If I add more GC, what does it do to the TM? It does. Is that going to work in our thermal cycler? It means we got to raise the temperature, right? Which means your enzyme might not be compatible for the thermal cycle. So when I want to activate the enzyme in the appropriate temperature range, if your primers aren't set up with the right biophysical properties, they won't go to the double strand or single strand situation when it's needed, right? 30 base pairs, right? You can go a little longer, but remember I mentioned when you go much longer, you start to introduce probability of other secondary structures, more loops, more problems, more unwanted populations, right, from laying down and binding, right? Avoid secondary structure. Lots of software exists. You could spend a fortune. Unless you're in a nucleic acid lab, there's enough freeware stuff to do just about anything these days, right? So, General approach, exact single base pair mismatched DNA thermodynamics. It's a mouthful. How does a mismatched piece of DNA affect its stability? G can bind to T, but do you think it does it well as C? Nope. Your body will tolerate it in some cases. But let's look what it does to the TM, to the melting temperature. All right. Oh, I thought there was an example of that one. Well, when you insert the T, you're going to wind up with a lower or higher TM. What do you think?
Lower.
It's going to be lower, right? We've taken away some of the stability. You're correct. So there's other calculators. You can use Gene Runner here, right? And it'll tell you the different TMs. We'll talk a little bit about mismatches at the end. I've got some real-world examples for you. You have fusion calculators, right? So each polymerase, basically each company that's going to sell you the different kits has their own little calculator. They've tried to make them a little more proprietary, basically, so you order their products. I mean, even at the end of some of these things, they'll be like, buy our stuff now. If it works, means to an end. All right, so what is this thing right here called? It's a plasmid, right? And what's the overall goal again? Make sure I've got you guys on the same page. What do we use these plasmids for? Typically recombinant protein expression, right? To manipulate the genome of something, to insert a protein of interest into the bacteria, or in my case, I often put them into different cell types to study them, if I want that cell to make ion channels, right? There's several components of the plasmid, right? You have an ORI, your open reading frame. You have a Lacan operator, so we can make it inducible. You have your gene of interest. And you have all these things up here. What are these? Anyone hear of BAM-HI, XO1, NDE? They're restriction sites, right? They're small little enzymes, think of as molecular scissors, that will cut specific sequences. You can cut and paste all sorts of sequences inside. This is the field of molecular biology, right? Making plasmids, making constructs, manipulating all of the nucleic acid information that's going to be used to be overexpressed or to be transfected or transformed into your cells. So when we do a transformation, right, we transform into bacterial cells. This is an agar plate. We take our plasmid, we amplify some up. Well, sorry, we can transform, basically put it into what we call a competent cell. Does anyone know what the word competent means in this case? It's ready for uptake. And why is it ready for uptake of a plasmid? What do we do? There's several ways you can do this. How do you make your cells competent? So you can use a chemical to do it, or you can use electricity to do it. All it's doing is poking holes in the membrane. So poking holes in the sarcolemma, the outer membrane of the cell. So there's these small little porous holes for the plasmid to go into. Okay? Compare DNA. Why is it harder to calculate melting temperature and dimerization of proteins? Ooh, good question. DNA likes to do what? Nucleic acids, are they happy as single strands in solution? They base pair, right? What about proteins? Do proteins exist completely in isolation? Yes, no, maybe. No, proteins interact, right? If we go back to the beginning, proteins I think of as the construction equipment inside the cell. They do things, they carry out functions. They can bind to things. How many amino acids do we have? A lot more than you do nucleic acids. Now there's a reduced number of torsion angles, but what drives most protein interactions? What types of interactions are there? You have hydrophobic interactions, hydrophilic interactions, electrostatic interactions, right? Many different types. All right, sequences of SH3 mutant. Okay, so if we're going to make a mutation to this protein, what steps would we do? First thing we would have to do is we'd have to design primers, right? So you can use an adjuvant web program. You're going to then do the experiment, right? So you design your primers, you're going to do your PCR. Once you've amplified your plasmid, you have to sequence it, make sure you made what you're supposed to, right? Sorry, that's do the experiment and get sequence results. And then check the sequence to see if the mutation was incorporated. All right? how do you sequence your DNA? Does anyone know? How do you sequence a plasmid? What do you do with it? Has anyone heard of a DNA footprinting assay? Okay, maybe we don't have time to go there, but it's an older technique. But long story short, most days you take your plasmid, you take a small amount, you put it in a tube, you send a primer out, and a company actually comes back and gives you a different set of reads, right? And based on the color and the absorbance, and please always check the chromatogram for your sequencing. Look at it, make sure you have a clean peak for each nucleotide there, right? Each one's usually in a different color. Sometimes you can get equal size peaks, right? And make sure the sequence is verified. All right. So, what problems can happen when you go to make a mutant protein, right? Well, step one is design your primer, right? I would actually introduce step 0 is look to see if the protein has some structural properties already determined. Look at the protein and see where the mutation you're making, right? And see what it would do. Then look at your primer. You got to make your primer, right? Primer could bind to itself. Primer might be too long and adopt too many other secondary structures. The primer might have multiple sites that it finds on the plasmid. How do you learn how to do this? You got to try it. I can stand here and talk to you all day about it, but until you try it and play with it, you won't learn it. It's just like if I were to teach you, tell you how to change your car oil. Until you physically go out there and do it, right? You're not going to learn it. What's great about this, it's not expensive. It's not a significant time sink. Play with it. See what you can do. Look at your favorite proteins. Look up some papers on new disease-associated mutations. See if you could make a primer that could modify the sequence of that protein, right? Maybe someday you'll encounter someone with money that wants you to, that you can play with, right? And, hey, I want to make a study of this protein. Okay, here's my methodology. Could happen. So, there's advanced computational tools for nucleic acids, right? DNA has a primary structure, your 4 bases, but it's far from simple. So what sounded simple in 8th grade of just AGCT is actually far more complex, right? Watson-Crick played a big role in determining some of the structural properties, but we don't understand it all, I will tell you that. And to leave you with a little bit of complexity to it, believe it or not, DNA doesn't actually stay base paired the whole time. It's actually a breathing component. Someone said there's an equilibrium constant, right? So DNA actually has a breathing or a base opening rate. It's actually dynamic, right? Base pairs actually form and reform on a certain time scale. Combining DNA and protein tools makes it possible to do a lot of advanced sequencing analysis. There are proteins that bind nucleic acids, right, during replication. There's all kinds of things that you can get into. Science is cool. So, with that, I want to leave you with a few examples of studying nucleic acids. So, this was actually my first manuscript I got involved with, and we were looking at MSH26. It's a repair enzyme. It finds mismatched DNA, okay? So they were looking at this. Now, it's a bacterial system because we couldn't study the human one. It was too complicated. What we were interested in is looking at different mismatches and how often they get recognized. So our collaborators figured out that different types of mismatched DNA get repaired sometimes better or worse. And again, this is important because if you don't repair a mismatch, if you accumulate too many errors, you can wind up losing control of the cell. It can become cancerous. Right? Broken DNA is one of the driving forces behind cancer. And so in this investigation, I characterized a whole lot of thermodynamic properties of DNA duplexes to understand how the sequence surrounding them affected their properties. What it turned out, when you put purines on either side in a certain spot, that breathing component altered, and it changed, versus whether you had pyrimidines. You can look at all the different K-ons, K-offs, predicted TMs, and stuff that we calculated. At the end of the day, oh, this is cool. This is the NMR spectra. So you can use, this is the one-dimensional NMR. You can go through and assign those amino protons that I talked about. You can look at the different stabilities. And what we found is that when you have the base pair lifetime, when you surround it with an A or a C on the GT mismatch, was much more stable. Here it was much more dynamic. And this gave room for the repair enzyme when it was more dynamic to shove an amino acid in there and recognize it and start to excise the damaged base and fix it. And therefore, we're able to say that some mismatches are more dangerous inside certain sequence contexts, right? This was important for understanding how DNA damage accumulates and how the body fixes it. So we can study the physiology of it. Not all DNA, as I mentioned, investigations are just about disease. Here we were looking at a treatment. As I discussed, we can use DNA and partner with RNA, and we can make this hybrid. What I told you before is that the DNA and RNA has a problem. You couldn't get it into a cell. So Barbara Shaw from Duke figured out that if we put these boron groups in place of an oxygen, a BH3 group, you could stick it in one of two spots. Remember, there's two oxygens on that DNA backbone. You have an R chirality or you have an S, depending which oxygen you substitute. One of them worked and one didn't, and they didn't know why. One protected it from degradation and allowed the antisense property to degrade, right? So you could put it in two things. So I was asked as a graduate student to characterize the structure of this. And was there something unique about the structure? So you can see your RNA on the bottom, your DNA on top. We modified this oxygen and put a BH3 group. It took me 2.5 years to learn the NMR for this. It was really a fun time. At the end, we were able to characterize this is the DNA sugar puckering. So the ones that were modified by the group, you could see here that the S modification had a large impact to this region where the R did not. You can see this altered part of the groove characteristics and changed some of the local phosphorus. So change the structure of it. You can go through and correlate this, and we even did some, where is it, some boron NMR spectroscopy. Right here, we actually measured boron, which is pretty wild, not something you typically do. You can correlate those boron peaks to other parts, and you could see here's the primary data where this is different. That boron was oriented in a different way. And ultimately, at the end of the day, I was able to determine NMR structures. You can look at the two structures, and what wound up being different is the decoration of the borons along the side changed the ability for the enzyme to grab onto it. And so the R enantiomer form was less effective than the S at accommodating the enzyme. And therefore, when they made a racemic mixture of these things, they had to spend the money to try and purify this, right? And it explained why some of their doses were more potent than others in terms of achieving an efficacy effect. So here you could see an overlay of my NMR structures where the S points inwards and the R points outwards, and you had this difference in bulge down in the backbone. So you can see here where the enzyme's trying to interrogate this, where the R is going to clash up inside here, and it doesn't accommodate for the enzyme to work. So RNase H couldn't degrade it. So I think that's about time, right? All right, with that, we're going to save some time for questions. I want to thank you all for your attention and for being here today. Any questions? So hopefully I've convinced you nucleic acids are cool. You can do a lot of different things. Not only can you change the sequence of your favorite protein, but you can study the nucleic acids themselves, right? You can study them for disease, fundamentals of cancer. You can study them for treatments, therapeutics, all kinds of stuff. Science is a big world, right? So, all right.
Okay, let's thank Chris again.
All right.